So this is how I will be talking. Throughout my presentation will discuss on viral disease, their significance in agriculture cropping system, understanding their molecular epidemiology and also etiology, how infections can be detected through different techniques including high throughput sequencing, particularly unbiased detection, and how this information can further be taken forward to develop the specific target detection tools which can then be used for management. So as far as viral infection of different cropping system is concerned, the ICPP master list as on 24, the latest one which is updated, which is edition of 13th and 9th edition, it recognized around 1,677 species which infect plants, and they are classified to 190 genera. So it's basically specifying that how many distinct group of viruses across the domains which can infect the plant species, including agri and horticulture crops, which are the crops of our industry. Now viruses, while they are so diverse, they have different modes of transmission, and they can be transmitted through vegetative propagation, they can be transmitted through insects, nematodes, fungus, even mites. They can also be transmitted through parasitic plants, contaminated foods, seeds, pollens, and also through root contacts as well as soil and so on. So this makes their management very difficult, and as far as our country is concerned, we have vast devices to infecting both animals and vegetable crops. If you mention here, you can see Asian leaf curly mopra, you can see yellow vein mosaic virus, infection, you can see recently the most important viral disease in temperate region, that is in apple, you see necrotic mosaic virus, you have different infections of Pistillin virus, nitrogen, and so on. So similarly, a lot of infection we see in the conditions, including both animals, field crops, and perennial crops. So why such infections are very important? They are very important because they causes a lot of losses in terms of yield, quality, and productive life of the plants. In animals, in the same life cycle, you see the losses in the yield and quality, whereas in perennial crops, the losses are even more pronounced because once the infection happens, the plant remains infected throughout its life cycle, and that viral load accumulates over the year. It also acts as a source of initial infection for fungus spread to the planting material or new crops which are planted. As far as the losses they cause in terms of the yield, particularly in perennial crops, many such studies have been conducted across the globe, both at economic level as well as yield losses in the percent, and you see that many infections like in apple, banana, you see infection up to 90 or sometimes 100% can be observed, and those can be correlated with the losses in terms of the yield. Now, when we understand the viral infections, it is very important to diagnose. And now, initially, we were diagnosing them at a particular species level, but now the concept has changed to the viral complexes, and mostly the diseases which are caused by the viruses are not because of one viral infection, but it could be because of mixed infection by many of the viruses. So, as you know, their complete cure is impossible. So, it is always important that we diagnose them well in time and we take the preventive measures, not for them to spread further in the plant population, and also we take control measures in terms of producing the clean stocks and keeping it clean, whatever way we can keep through certification or regulation. So, as far as their diagnosis is concerned, we can have either non-targeted method or we can have targeted. When we say non-targeted method, it could be through symptoms, it could be through transmission, which can be undertaken through biological means or through indicator host, It could be through structural characterization of the viruses. Generally, the most common is electron microscopy, which we do. So you can see whatever viruses are present in a particular sample. Nevertheless, many times our two methods they miss many of the viruses because of the less load or less concentration in the tissue or not improper sampling we do. So we have many other methods in the second line, which could be the targeted one, where we develop the specific diagnostic assays to detect them. So it could be through ELISA, which is a serological based method, or you can have any molecular detection method. It could be PCR, PCR, isothermal assays, or so. Very recently, we are now using for last more than 1.5 decade. Plant biology has witnessed a paradigm shift in detection of viruses associated with a particular disease, and we have high throughput sequencing, which is mainly employed for detection of the associated viruses. So we already mentioned that the other commonly used method has their own limitations in terms of specificity and sensitivity and limited coverage. So in the era of omics, we employ high throughput sequencing for the non-targeted neutral detection of the viruses, where we sequence the entire nucleic acid in sample where we want to see what are the viruses associated. So high throughput sequencing has a specific advantage because here we do the parallel simultaneous sequencing of the multiple DNA fragments. Mostly you take RNA as a starting material in case of all viruses, whether they are DNA viruses or RNA viruses, because all viruses have their transcripts in the once they start replicating in a particular cell. So you can use the RNA, total RNA, and then take the cell as a starting material, and you can convert it to cDNA and then followed by parallel sequencing on either of the platform. Mostly we use shotgun approach, and then lot of massive data you generate, and these data can be further analyzed to see what are the viruses present. So it basically provides a good advantage because you can identify the potential pathogens without having identified. You don't need antibody, you don't need any sequence information which is otherwise required in nucleic acid detection method. Here you can detect everything what was present in the sample which you use as a starting material. It is rapid, comparatively rapid, accurate and efficient, and you can see the entire virus. This is at the later stages that you correlate it with a particular infection and you correlate it pathogenically, but at the initial point of time you will be able to decipher what are the viruses which are present in the sample. The other advantage of this technique is that you can identify both known and unknown viruses as well as characterized as well as uncharacterized viruses, including understanding of their diversity, evolution, and also molecular epidemiology. Even a very low titer viruses can also be detected in the sample. That is again an advantage. So how we basically do, we take a plant sample, we subject it to high quality total nucleic acid isolation, and this DNA or RNA we take configuration of the cDNA libraries. You label it with the vectors. Vectors are basically the known sequence labels, and then you make libraries and you sequence. Once the data is generated, you analyze it. So you analyze it in a way that you would like to see what are the viruses present in a particular sample. And so here you need a massive data analysis tools, which is basically a computational tool using in-house or whatever validated pipelines. In most of our studies, as well as globally, we use short-read sequencing to analyze the virus of a particular sample, and this is generally done on even as you can see from the right side of the slide, that mostly Illumina gives you very good amount of data which can be further assembled to see the viruses. So how such techniques has been used by us to analyze the viruses. I'll take a few examples from our own studies. So we started working extensively on this particular technique, and based on our experiences, we have developed, validated, and optimized some workflows to analyze the virus in a particular sample. So you can see mostly we take the sample, so it could be a symptomatic sample as well as asymptomatic samples which we want to see. You subject it to high quality RNA isolation. You check the quality of the RNA, then you go for the library preparation. Once the library is prepared, you subject it to
only 5 minutes to go.
Only 5 minutes?
Yeah.
So high throughput sequencing, and once the data is generated, you analyze it to see the viruses. So using this technique, basically we have identified number of viruses and also understood their epidemiology. One of our studies, which is published in Physiological and Molecular Plant Pathology, we studied the grape nurseries and both rootstocks and signs and undertook their viral studies to see the viruses present in there. So once it is done, we could identify at least 12 viruses present in either of the rootstock and sign. Once such viruses are identified using Illumina platform, we wanted to detect their prevalence specifically in mother banks of rootstocks and signs. So if you see this table, just to highlight the major findings, you can see that there were some viruses and viroids which were consistently present in rootstocks, whereas others were present only on the signs. So basically it shows that how viruses have moved from the rootstock to the signs. In addition to understand their epidemiology, we were also able to identify some of the novel viruses like GLRS, grapevine microvirus, and one novel viroid, that is grapevine yellow speckle viroid. Similarly, in case of apple, we had developed a pipeline and we trained on multiple data sets, and we could decipher the infection of new viruses, including the virus, that is citrus contained domesticated virus, and apple replicating virus, which is the virus. So, in the same time, we had deciphered the virus of the field, identified the viruses associated with the symptoms which we observed in the field. We also deciphered the virus of the plum, similarly peach, and many more crops which we are doing now. So, despite having this advantage of the high throughput sequencing, we can still have some issues. If you have a sample bias, means you don't sample proper tissue, which should be subjected for sequencing before RNA isolation. So, also degradation could be an issue, because this technique is very sensitive. So, contamination is again an issue, as well as some bias. So once the viruses are identified using high throughput sequencing, we can take the viruses which are economically damaging, and we can develop a specific diagnostics which can be used for targeted detection. So our group here, we have been using isothermal detection assays, mostly R2, and it's a recombinase polymerase amplification assay. The assay could be performed at 37 degree isothermal temperature in 30 to 40 minutes. And using this particular assay, we have developed wide array of the diagnostic assays for detection of the viruses as well as virus-like agents, including those of lateral probe CPPS. So this one we recently published in Drop Protection, and this is for detection of candidates like bilateral CPPS. And the advantage really of the assay is that you can detect the infection in the true plant extract, isolated, prepared in specific buffer or particular solution. Similarly, we have developed our duplex IFCPAR simultaneous detection of two DNA viruses in banana, that is banana stick MY virus and banana stick Coimbatore virus. So we developed a number of assays, at least 5 dozen of viruses, and many of them now we are using in the national certification system participated labs. So with this, we have completed detection, both neutral and specific detection. I'll take a few minutes just to brief about the management, how we can use different recent techniques for management of viruses. So our group is also using topical or endogenous application of DSRNS, which is in vitro synthesized for management of the viruses. And recently we have used this technique for management of one of important virus, actually that is chili venom virus. So we use the DSRNA against three genes, that is the silencing, suppressor and assembly. And this application can prevent the infection of this virus in the lungs. Similarly, we are also using CRISPR-Cas9 for editing of the potential or known susceptibility factors. So our group has started working on rice, and our senior group here in IRL, they have a long working experience on using this particular technique in tomato. So their target is pilota, which is one of the known susceptibility factor, and which helps the establishment of the viral replication in tomato. So this pilota, which is a susceptibility factor, at a particular site, that is position number 16, they use a CRISPR-Cas to mutate it from valine to the glycine, and once these mutants are developed, these mutants of tomato are resistant to tomato viruses, particularly the virus and virus. So this is just to conclude my presentation that techniques like high throughput sequencing will help you to identify the viruses at a neutral level without having any bias. Once you identify the viruses, whatever important group of viruses are present, you can develop diagnostics, and then this can be used for regulation or certification program. As far as management of the viruses is concerned, we now have some new techniques like DSRNA-based management as well as CRISPR, which can be used for management of viruses, and we can use if we have susceptibility factors known in a particular host virus ecosystem, and they can be managed. So I would like to thank all my funders as well as collaborators for this work, and also TNAU for inviting me to talk briefly on the work which we have been doing. Thank you.
Long live India and France friendship.